Okay. Okay. So I think we should be about an hour, like maximum, after this last one. I think we'll probably finish still around four-ish, maybe a little bit after, but I think we're doing reasonably good on time now. So yeah, here I kind of just go a little general into soil health, but I just kind of want it's kind of a bit of a piecemeal. We're going to just touch on kind of lots of different themes. We've kind of had quite a focused discussion today on the plant nutrition and the foliars. There's a lot of other stuff to talk about, and there's a lot of other things that I often talk about and can talk about. There's a lot of other kind of topics. So what I kind of wanted to do here was just stitch together a little sprinkling of some of the other pieces of the puzzle. I think the soil topic is, of course, is a big one. There's many kind of aspects to that. So I kind of thought we'd just weave a few kind of different kind of concepts together into a theme here, and kind of touch on a couple of range of different areas around soil health related. Just while I remember, that's my website there if anyone's interested, integratedsoils.com. I do have some like resources and things like articles I've written or media pieces and things that have been written up about some of my talks and things. So if anyone's looking for some more reading material, you'll find stuff that you've heard a bit about today. There's some other details there. I've got some articles on nitrogen inhibitors and other aspects to nitrogen and things on there, and it's kind of like other stuff. You'll find a bunch of stuff there. So if anyone's interested, they can also have a little look there for some other material if you'd like to dig a little deeper. So let's dive into soil health. We're going to touch on a bit of soil health from the bottom up and top down. I'll explain what that means. And then we're going to kind of move quickly into just a bit on kind of covers, and a bit more about diversity. There's a couple of little things I wanted to mention about diversity and some of the kind of intercropping, kind of root to root kind of interactions, just some kind of cool things in that space as well. Just a little bit kind of that links all back into that kind of broader diversity discussion. We did discuss diversity with through that specific lens of pest and disease pressure. So I just wanted to maybe branch out into some of the other benefits of diversity. So this kind of classic idea of soil health is the chemistry, the physics, the biology, and I do really appreciate the three-legged stool analogy. I think it really is a great one because obviously it points out that if any one of the legs on that stool was wobbly, loose, etc., it's of course the stability of that soil is going to be not so great, and equally the function of your soil, the functionality of your soil is also going to be limited, not to its full potential. If you're not focusing in on optimizing the chemical parts of the soil, the nutrients, the pH, the biological, those organisms, of course, the micro to the macro, and some of the structural properties, the aggregation, the infiltration, the soil structure, and they all work together. I know that soil biology is the buzz at the moment and the buzzword, and we're all really interested in soil biology, and rightly so. It's the black box. It's the one we know least about. It's the one we're filling the gaps in now. So it's only fair that we're all very interested and excited by maybe the potential that can come out of this, how this can help us. But those organisms, they still have to live in a physical world, in the soil physics, in that structure. We still need good structure for them to live, a good home, and they still need good nutrients availability too. We talk about plant essential nutrients, macro and micronutrients for plants. Well, the microbes also need nutrients. They also, just like we do, just like livestock do. Everyone needs also those nutrients as well, just like the amino acids we were talking about earlier. So we've got to have good nutrient supply for them too. So I do really think at this point that they are all of equal importance. I know biology drives so many of these functions. Biology can restructure and improve the physical condition. Biology can drive nutrient cycling. Of course, they can do many great things to optimize those other two legs. But it's a feedback system. They're all kind of working together as a whole. And of course, that's why when we kind of tease this out into more layers of detail, we talk about some of the particular things or the soil health metrics, things that we can measure to try and manage under those three different categories. So, of course, we're looking at things like the texture of the soil, the structure, what's that aggregation, what's our infiltration like, some of those basic in-field tests like the infiltration, which gives us an indication of the porosity and some of those structural properties. I think they're fantastic, great, easy things we can test and measure in field. So, do we have any signs of compaction or surface sealing? All of these things are things that we can try and measure and monitor to track. What is our soil physical condition like? Are we improving it over time? The biology, of course, soil organic matter, soil carbon. I mean, that kind of sits in the middle. Carbon influences the physics, the chemistry, and the biology, but I put it in there. It's the main food source and the house and the home for them, particularly those microorganisms, so we can measure them. Who's there? How active are they? What's the diversity of them? Through to some of those other soil insects and earthworms, of course, doing earthworm counts and some other indicators of their biological activity, measuring some of their enzymes, their exudates, those microbial metabolites and things. They can be indicators of how well they're functioning. And then the chemistry, of course, the pH, the nutrient availability, what kind of storage do we have, what's the available fractions, that kind of thing. So, we can kind of track all of these and try and break that kind of big picture of soil health down into specific things that we can measure and manage to try and kind of piece together our progress and how well we're functioning. So that's kind of the component parts and that we try to measure and therefore improve. And that's that kind of really, when I say bottom up, that's kind of really what I mean. We're talking about soil health from the bottom up. We're trying to measure these things to try and change these things, try and improve these things. If I make my soils healthy, if I fix these things, if I optimize some of these, if I make my soils healthy, then healthy plants will grow. So we're trying to change the soil, improve the soil towards this kind of improving plants' health or production. That's kind of a little bit where that angle is coming from. But of course, as plants grow in those soils, as they grow in that physical world, get those nutrients interlinked with that biology, they get all the resources they need to photosynthesize, to grow, to do what they do. And of course, as we talked about, they breathe in that carbon, fix that into their bodies, but some of that carbon goes down to the below ground, down into the roots and down out as those root exudates. So plants are not just breathing in that carbon, they are also releasing carbon, bringing carbon into the soil. They are a pump bringing carbon from the air down into the soil. And when they bring that carbon down into the soil, they are bringing the energy into the system, the fuel, the food, to drive so many, especially biological processes, yes, feed those organisms, but also many of those root exudates, there's some of those organic acids in there that are solubilizing the nutrients, some kind of polysaccharides, chain sugars, sticky things that also help to glue the soil particles together, improving the soil physical condition. So they do lots of different things. Root exudates are a diverse array of compounds. So the plant really is bringing carbon from the air and it's bringing it down into the system, which is the fuel, which is the food. It's really the key energy to kind of drive a lot of these processes. And of course, it is especially important for that vast underground world of the soil biology, all the way from the micro, the bacterias and fungal type things, all the way up to our larger little protozoa animals and upwards to the micro insects, macro insects, earthworms, etc. All of that little community of life we often call the soil food web. And of course, it exists in the soils in incredible diversity and vast, vast numbers, incredible diversity. And with that diversity comes a great diversity of function. They can do so many things in such diversity. That's why there's great potential in soil biology from the micro through to the macro. It's because they all are different. They're so very different. They all do very different things, and they can really bring multifunctionality into the soil. They can really help the soil in many, many different ways at many different scales, spatial scales, again, micro to macro, and through a whole range of different modes of action. And that's why they're very valuable to us. It is that genetic diversity in which they exist that means they bring many tools to the toolbox, much potential to kind of do all sorts of things for us. And when we talk about those root exudates there, of course, it's fine. We talk about the sugars. Oh, the plant's photosynthesizing and sending those sugars down to the roots. Of course, there's nothing wrong with saying that in a general sense, but to use the word sugars to describe root exudates is a gross simplification of the true and intricate nature of what are root exudates. Root exudates are hundreds, if not thousands, of different compounds that the plant produces through photosynthesis first, but then through that secondary metabolism, metabolic pathways, that secondary metabolism, to produce all of these other different bioactive compounds and substances. Now, the bulk of the root exudates, yes, they are sugars, carbohydrates, all sorts of different sugars, and amino acids. We talked about amino acids. The plant, it'll release a lot of those. And again, why would the plant do that? Those are those highly efficient forms of nitrogen we were talking about earlier. But again, why would the plant give away sugars either? Why would it give away those valuable amino acids? It's doing that to invest in those organisms. It's not a waste. The plant is investing in that biology because that biology is so effective with its multifunctionality at getting nutrients, at restructuring, at protecting the plant from disease, doing all those things. So the plant is investing in those organisms. And that's why it will also give away a lot of amino acids as rutexudates and organic acids, some of those acidic compounds to solubilize the nutrients, etc. So the bulk of your rutexudates, yes, it is sugars, carbohydrates, amino acids, and organic acids. That is the bulk of the rutexudates. But underneath that, we have a whole range of very specific and very unique to different plant species, even to different varieties, a lot of very specific biochemicals. And later on at the end of the session, we're going to have a spelling bee. I'm going to ask you to pronounce and spell some of these compounds. Don't worry, I can't pronounce any of them either. But what we know about these other biochemicals is that they're not just about raw feed. It's not feeding, it's not energy per se. These biochemicals are what we could call the on-off switches. These are the biochemicals that are recruiting certain microbes, activating, waking up, recruiting certain microbes, but also putting others to sleep. No, I don't need you anymore. I've finished that growth process. I've done that. I don't need that nutrient anymore. I've moved on in my developmental stage. I need something else. I'm going to wake up some other organisms now. So that's what a lot of these biochemicals they are. They are actively turning on or turning off microbes. I don't need you anymore, or I do. And it's some of those really can be as simple as some of those food sources, some of those rutexudates are simply preferred food sources for some of those microbes. So certain microbes, particularly like citric acid, for example, so all the microbes that like that, they become very active and they start to kind of feed on that, for example, or some of these kind of specific chemicals that kind of break their dormancy or trigger them to wake up, for example, or shut them down, induce a dormancy. So that's really the essence, and that's how the plant is regulating its microbiome around that root system. And that's why, coming back to my earlier point about putting microbial products onto the seed or into the furrow, this is where I think they're most appropriate, because then you let the plant do the rest of the work. Just put the organism there, put a diverse inoculum there. Yeah, sure, I'm a fan of compost extracts. I think they're good because they're very diverse. So let's just put a diverse inoculum there, and you can put that same diverse inoculum under wheat, under corn, under soybeans, whatever. And corn is going to release different sets of rutexudates and recruit and activate the organisms that it wants and needs, which will be different from the ones that the wheat does or cereal, or different from our pulses. So you let the plant do the rest of the work through these specific rutexudates that it will recruit the organisms that it requires at all of those various growth stages. So that's why I like the diverse inoculum of the extract best. Just put a big old diversity out there. And sure, your maize is probably not going to recruit half of those, but that's going to be different from the half that the wheat's not going to recruit, etc., etc. So they're going to take the bits that they want and need, and that's specific to those specific plant species. Our job is just to put a diverse inoculant there. And yes, again, at the end of the growing season, they may be dead after that. That doesn't mean that they weren't valuable. So there's a lot more complexity and nuance. Rutexudates are highly dynamic. The plant changes this all the time. If cold temperatures are coming, heat waves, if it's under pest attack, as we talked about, this is a dynamic process, and it will change the cocktail, the composition of those rutexudates regularly through the course of the growing season in response to the environment, in response to its changes in developmental stages, etc., etc. So that's that idea then. That's kind of then plants photosynthesizing from that kind of then the top down. And here we kind of bring this merging of this bottom up and top down idea. When we think about, this kind of classic saying, I'm sure many of you might have heard this one, feed your soil and it will feed you. This is like really brings together this idea of soil care, soil nurture. I want to feed my soil. I want to take care of my soil. And when I take care of soil, when I make healthy soil, when the soil is healthy, then healthy plants will grow. So it puts this focus on the soil. I'm going to make my soil healthy. I'm going to improve those physics, the chemistry, the biology, work on those properties. Feed the soil, put some carbon into the system, feed it, and good things will grow. And that's that kind of then that bottom-up idea. So, of course, we're encouraged to maintain stubble retention, maybe make some compost, use some manure, apply organic amendments, feed that soil, look after that soil, take care of that soil. And that's your kind of your bottom-up approach. And then we have this idea that, well, okay, but plants also grow in soils, and as plants grow and they photosynthesize and they put all that carbon down into the system, they also begin to change soil as well. It's not just that plants are an expression of a healthy soil and make my soil healthy, and healthy plants will grow. As plants photosynthesize and pump all those root exudates down, they begin to change the soil. They change the chemistry, the physics, and the biology. So they also begin to change that soil health and the soil properties. And that's captured in, of course, that beautiful quote here from Ray Archuleta: soil without biology is just geology. So here we now are tapping into this top-down approach, this top-down idea that says, well, actually, it is plants that photosynthesize, that bring carbon into the system, that put that carbon into the rock, into the parent material, that actually drives the process of soil formation. Without plants putting carbon into rock, into parent material, we don't have soil in the first place. If we think about this context of soil formation, how do soils form? They begin with geological activity, rock, parent material, geologic upheaval. Well, what happens is things start to grow on it. Biology starts to grow. Micro things, mosses, lichens, algae, bacteria. And the micro things, biology, they grow first. And they attack that rock, and they start to strip minerals, nutrients out of that rock. And they grow and die and grow and die and they grow and die. And as they die, they are depositing their dead bodies, carbon, into the rock. So now we're beginning to mix mineral, inorganic from the rock, with carbon, organic from the biology, from the living things. And so we begin this process of soil formation because of life, because micro plants like algae and other organisms begin to grow there. And of course, as they do that generation over generation over generation, over time, they start to form topsoil. We begin to form what is soil, a mixture of rock, mineral, inorganic, with organic plant life biology. And so they begin to mix and interact, and we drive that process of soil formation to such an extent where higher plants can now move in, those pioneers, the weedy type plants, early successional plants, annuals, etc. And they again start to really accelerate that process. They grow and die, getting nutrients out of the soil into their bodies, dying and returning the carbon, the organic material. And then they, of course, are increasing that soil formation as annuals through to that point where we cut into perennials. And when we cut into perennial plants, here we really accelerate that soil formation process because perennials invest much more heavily in roots, in below ground carbon. Perennials are coming back next year. So they invest in roots. They invest in below ground carbon for storage to help them recover the following spring. Whereas an annual, it's not going to be here next year. So it puts everything into above ground, into reproduction. It doesn't really grow many roots. It invests in above ground and seed production quickly. Perennials, they invest in roots below ground. So when we switch to perennials, that's where we really drive now to accelerate the soil formation process, particularly when coupled with livestock and grazing, as you have done here on your beautiful prairie soils. We really accelerate this process onwards through to succession and forests and etc., etc. So the point is that we wouldn't have any of this if we didn't have life interacting with rock. It's biology that is driving the process of soil formation, and we can extend that to say, well, it's biology that drives a lot of changes in soil health. And of course, that's why if we look at the moon and Mars, we have geology, we have rock, but we don't have soil. We don't have the life. Okay, so it's the biology. And when we say biology, it is absolutely plants are key because they are the ones getting the carbon out of the air and starting the whole chain reaction. It is plants first, or photosynthetic microalgae as part of that. But it is plants who have the special ability to bring carbon, the stuff of life, the biology, the food, the fuel, the energy, and mix it in. And then we drive, then of course the microorganisms, the bacteria, the protozoa, the insects, the earthworms. Then all of those other things come because they feed on that carbon that the plants bring in. So this is our top-down view that says that actually without plants, without photosynthesis, we wouldn't even have soil in the first place. So these are our two world views. Bottom up, I've got to make myself healthy. going to make healthy plants grow versus, oh, well, actually, if I just grow plants, if I make my plants healthy, they're going to grow into that soil and drive not only soil formation, but also soil health restoration, changes in soil health, etc. And that's where they kind of intersect. And, the simple example of the top down, of course, is like cover crops or green manures. Like, what is a green manure? It's a plant that we grow to improve the soil, to increase the soil, to improve the soil properties. So plants change soil as well. So not only do healthy plants come from healthy soils, it's also that healthy soils come from healthy plants. And what is a healthy plant? Well, my definition of a healthy plant, or not definition, but my thinking of healthy plants is nutrition, optimizing plant nutrition. So you optimize all of those photosynthetic pathways we talked about, and diversity, plant species diversity. So do we optimize plant species diversity? Do we optimize plant nutrition? These are the tools to make healthy plants that will then drive changes below ground in that healthy soil. And absolutely, it is what is happening below ground that is so key to driving those changes. It is roots. Roots more directly drive the changes in soil formation or soil health restoration. It is roots that much more directly drive this. So it's key to be thinking about below ground carbon. And of course, that's one of the challenges with production agriculture, is that we generally are trying to encourage above ground biomass, because that's yield, that's what you harvest, that pays the bills. And that comes at a trade-off to roots. And this is one of the reasons why we've seen a bit of soil health decline in the last decades with intensive agriculture, because we bred varieties to be very productive, meaning they put a lot of carbon above ground. And we harvest that. And that means that's a trade-off. They don't have as much carbon below ground in those roots. And it is the root carbon and the root biomass that really drives some of these kind of changes to soil health. So we've got to be thinking really critically about roots. They are the key aspect to soil health. So yes, it's chemistry, physics, and biology. That's kind of bottom up. But actually, soil health really looks like this. It's got plants in the picture. As plants grow, as their roots penetrate into the soil, yes, they change chemistry, they change physics, and they change biology. Plants will change the soil as well. And that's your top-down view that says that soils are also an expression of that plant health. So this is really how we should be looking at soil health, conceptualizing for soil health. Plants and soils, they go together as one, of course. And when I say plant health, for me, that means optimized plant nutrition and plant species diversity. And of course, when we do that, when we optimize that kind of process, there's examples here of kind of changing that soil chemistry. You do have a lot of nutrients in your soil. There's a lot of opportunity to improve nutrient use efficiency, to use less fertilizer, and we talked about the folio aspect to that already. But also, you have a lot of fertility in your soil that you can just use, that you can leverage what's already there. Now, in a light sandy soil, you don't have so much potential. But if you've got a medium soil, clay soil, certainly heavier clay soil, you have lots of nutrients that are there, that are present, but they're just locked up. They're just not available. Only a very small fraction of the total nutrients is actually soluble in the soil water available. You've got another kind of fraction here, the exchangeable or the medium-term supply. That's the nutrients kind of sitting around on the soil colloids here, the exchangeable forms. But then you've got everything else, the total stuff that's still embedded in the mineralogy of the soil, some of those nutrients that are locked up. You have lots there. Maybe not in a sandy soil, but the other soils, absolutely. And you've got a huge potential to just use this. And when you do a standard soil analysis that's just going to tell you what's more of the available fractions, you're not getting the full picture. And this full picture can be ginormous compared to this fraction. It could be 10 times more, minimum, or more. There are big reserves in soils, in certain soil types, where you can just leverage and unlock this. And that's where biology, plants, photosynthesis, feeding organisms, the diversity of those microorganisms have lots of tools to then unlock and make use of this. And so that's how we drive that nutrient cycling through plants. And therefore, this is our other opportunity. Hence, this is the point of the cover crop. What's a cover crop? We're trying to get fertility out of the soil, bring it into the biomass and return it into an organic form as a nice multi-litter layer on top, which will break down, which will recycle those nutrients from the soil back for production. And that's really, of course, what we are essentially trying to do with our cover crops, with our green manures. So that's your then top-down, using plants to kind of drive some of these changes. And I think that's captured really nicely, this idea, in this quote. This is from a Canadian rancher in Alberta, Steve Kenyon here. Modern agriculture grows plants from soil. Regenerative agriculture grows soil from plants. So there's our kind of bottom-up view, and here's our kind of top-down view. Therefore, if we want to improve soils, we can improve the management of plants. And again, like for me, that's nutrition, that's diversity, and I guess I should add, and grazing management, how we use the animal to manage those plants in terms of the grazing. So of course, that's a big part of it from a grazing context too. So I think that captures really nicely these two kind of paradigms or schools of thought there. So let's kind of move a bit into then this point about the roots. I touched on roots there and their direct role in soil health or building soil organic matter is another part of that discussion. When we think about plants and the residues in which they leave behind, certainly cover crops, but even within our other cash crops as well, or again pastures, we have kind of three components to plants. We have the above ground biomass, the shoots, the below ground biomass, the roots, and then we have those exudates. This is the three pools, the three fractions of the carbon, the photosynthetic carbon that got into the plant. This is our three fractions of carbon, that energy that can then be integrated into the soil and be used to drive some of these soil health changes. So it's going to be different percentages and balances of all three of these. Now, of course, with production agriculture in the last decades, we did a really good job at increasing yield. We did a great job at that. And yeah, maybe we're plateauing a little bit here or there in various contexts. But, since the Industrial Green Revolution, sure, we have done a really good job at increasing yield. Now, shouldn't that mean that we've been increasing all of the amount of residues and carbon and photosynthetic potential coming into the soils? Shouldn't that mean that we have been increasing our soil organic matter? All of that extra yield, driving all this extra residue decomposition, all of this building soil organic matter? Shouldn't that be the case? I'm pretty sure that our soil organic matters, since intensification of agriculture had generally declined, gone a bit the other way. There are some exceptions to that, for sure. But generally, we've seen organic matter being depleted in our soils in recent decades, despite the fact that we've been increasing production, increasing yields. And that's just for the very simple reason that I mentioned there earlier, is that when we increase yield, we increase the shoots, the above ground biomass. And if you want to build soil organic matter and build soil carbon, the key thing you need to do is grow more roots. It is roots and not shoots that build soil organic matter. And we have lots and lots and lots of studies that have looked at this and integrated shoot material into the soil and root material into the soil. And as they decay and break down, roots make a greater contribution to soil organic matter than shoots. And one of these studies here kind of was a review, and it reviewed a heap of other studies, and they came up with this figure of about 5 times. It's about 5 times more, roots are 5 times more efficient, more effective at building soil organic matter than the root material. Now, part of that is just simply the location of roots, where roots are. Roots are down below in the soil. Shoots sit on top of the residue, and that's part of your soil armor. That's actually not how you're going to build soil organic matter, by having all of that residue on the soil. It's good for other important reasons: soil protection, moisture conservation, insect habitat, earthworm food, etc., etc. Yes, it's great stuff, the mulch, the armor. And it helps protect the soil to help what happens below to function better. But the main driver of soil organic matter building is root material. It's below ground carbon. Below ground carbon is the key here. So, of course, when we increased yield, we increased above ground biomass. And when we shifted from perennials, these beautiful prairie grasses that we had here, and we shifted to annual agriculture, as I mentioned before, that switch to annuals means that annual plants allocate more carbon above ground. So we lost the perennials that allocate more carbon below ground. So the switch from perennials to annuals was a big first step there. Secondly, we bred varieties. We bred our annual varieties to be higher and higher and higher yielding, meaning more and more and more above ground biomass, and that came as a trade-off to roots. So modern varieties grow smaller and smaller roots these days compared to some of those older varieties. I'm sure some of you have seen that with some of the einkorns and Emma wheats and some of those kind of other things, for example. So that was another one, breeding. And then lastly, nitrogen. Nitrogen fertilizer is really good at increasing above ground biomass. And I'm not sharing the studies, I'm just summarizing for you today, but I do go into this in more detail in other presentations. But nitrogen drives yield, which drives above ground biomass. Nitrogen doesn't really increase root biomass so well. A little bit it does, but not really. And too much nitrogen definitely has a suppressive effect on root biomass. So these were the three factors: the shift away from perennials, modern plant breeding, nitrogen fertilizer. All three of these things have encouraged more yield. And that's good for the economics, but it has also been a decline in soil function, soil quality, soil organic matter over time because of the less roots. So roots, really, really important, but it's particularly below ground carbon. Because what about those root exudates? How do they fit into the discussion about building soil health changes or soil organic matter? And here's a great study that was looking at this, trying to tease apart the breakdown or integration of root litter versus root exudates and their contribution to soil organic matter. So, evidence for the primacy of living root inputs. Living root inputs means root exudates, not root or shoot litter in forming soil organic matter. Recent theory suggests that living roots exert a disproportionate influence of carbon formation, soil carbon formation, but few studies have explicitly tested this by separately tracking litter residue integration versus exudate integration into the soil organic carbon pools. And they do this experiment to kind of track these two things, tease them apart, and we show that those living root inputs are 2 to 13 times more efficient than litter inputs in forming both the slow cycling and more fast cycling, more slower stable and faster cycling carbon pools. So it turns out that root exudates may be even more efficient still, these liquid carbon sources versus the root litter inputs. And that comes down to not solely this. This is kind of an emerging idea here, so there's kind of, again, knowledge gaps, but where we currently kind of sit on thinking about this is it's a simple idea of carbon use efficiency or digestibility. And any of you cow people out there who understand digestibility, it's exactly the same concept, digestibility to the microbes. So when you're a little bacteria and you're fed this delicious, highly digestible, easy to assimilate, small, simple sugars and amino acids, etc., this liquid carbon here is highly, highly digestible. And microorganisms in the soil can immediately make use of this and feed on it and multiply and grow. Our microbial friends over here, however, unlike our cattle friends that have mouths and teeth and multiple stomachs, a bacteria alone is kind of looking at this stuff on the soil thinking it's a bit hard to chew. It's a bit hard to digest. So, of course, microbes don't have mouths. So how do they digest? They excrete all sorts of digestive enzymes externally. Digestive enzyme meaning lots of scissors that kind of cut this up. They cut up this complex lignin. It's difficult, it's highly indigestible, not easy to break down. So they have to use chemicals, digestive enzymes externally to cut. And so they start breaking that down. So this residue is going through this continuum of decay from complex stuff through to smaller. And once it gets small enough, fully digested small pieces of carbon, like what we had originally here all along, once we break this down into the small stuff, finally now our friends over here can ingest and start feeding and start growing and start multiplying. And our microbial friends over there feeding on the root exudates are long out of town. They have been multiplying for ages. They've been multiplying, reproducing, growing for ages already because this was immediately available and highly digestible. Meanwhile, our friends over here have to wait, digest, wait for these scissors to cut it up, and then they start growing. So because of this higher carbon use efficiency, the root exudates are more efficient at feeding biology and growing those organisms and feeding them. This is why the root exudates are the preferred food source as I mentioned earlier, to feed soil biology. And that's important. The digestibility of these two carbon sources is important because, as we now know, when we say soil organic matter, we used to think that that was mostly all plant material, decayed plant material. But now we know that soil organic matter is a mixture of decayed plant material, crop plant residues, etc., and dead microbes, microbial residues, microbial waste products, microbial necromass, dead microbes. And so it turns out that as microbes grow and die, when they die, they very effectively, they stick to soil particles. And when they stick to soil particles, they form a very stable fraction of soil organic matter. So soil organic matter, again, we thought it was mostly all plants. I mean, it does come from plants. Plants are the photosynthesizers. But once that plant gives the carbon in a digestible form to a microbe, that microbe eats it and turns it into microbe. It's no longer a plant anymore. When that little amino acid is eaten, it was from a plant, or as that sugar or whatever is eaten, now it turns into a microbial body, a microbial something. So now it's microbial. So once we get carbon from plants into microbes, then we can stabilize that carbon through this dead microbial bodies, these necromass. And it turns out that necromass makes up around about 50-odd%. 50-odd% of soil organic matter is of microbial origin. I mean, originally plant origin, yes, but it is 50-odd% microbial origin and about 50-odd% plant origin. So if you want to optimize both of these pathways, which you do, you don't want to just focus just on one or the other. We all want to improve both. It works better if we kind of have two highways to run down. More of us can get down there. If we want to optimize both, this just simply means that, of course, residue protection and stubble retention and keeping litter and things on the surface is important. That's just going to go through that bit slower decay plant pathway. But if you want to optimize the microbial pathway, you have to maintain living roots, because then you have root exudates. And those root exudates are highly digestible and will most effectively form the microbial necromass ultimately. So if you want to optimize both pathways to building soil carbon, you've got to do a bit of both of those. And that's where the soil health principle, and the science is catching up to this, soil health principle of living roots, is now very much supported by some of the latest thinking in soil organic matter science. This paper is from 2021. Okay, so that's just green cover, living roots. That's nice and simple. Okay, nice and simple. How are we doing on time? Okay, so that segues into things like covers and companions. I'm going to just say a little bit about plants and a bit about the diversity thing to kind of round this off. Covers, companions, they're great for lots of reasons, not just building soil organic matter, but also, for soil protection, including the function of that soil, building the soil organic matter as we talked about. They also help with water, so water infiltration, drainage, preventing nutrient sediment losses, improving water use and access. Covers, they help with great things from an agronomic point of view, like increasing soil fertility, cycling nutrients, improving those nutrient use efficiencies, leveraging that fertility in the total pool. Great for producing forages, of course, managing weeds, and the ecology part that we touched on too. Habitat creation, maybe having some pollinators, encouraging predators, other good biodiversity in the farm ecosystem, and improving some of those kind of general ecosystem services of the farm at whole. So plants help to do all these things. Keeping the soil covered ticks all these boxes. And of course, soil organic matter is just one of those. But which plants do we use? Well, it depends what the goal is, hey? Are you trying to increase nitrogen or phosphorus liberation? You're trying to capture nutrients, cycle nutrients, prevent nutrients from leaching. You're trying to suppress disease, brassicas we talked about. We're trying to encourage pollinators, so we need flowers for that. Are we trying to suppress weeds or build soil organic matter? Okay, building soil organic matter, we want to grow more roots, nutrient capture at subsoils, grow more deep roots, capture some of those nutrients, cycling them around. Weed suppression, we want high biomass, quick growing, smother them out, maybe some of those allelopathic chemicals in some of those plants, or compaction alleviation, again, deep roots, we want deep roots to get down there. So what plants do we use for kind of some of these changes in soil health and restoration? Well, it depends what you're trying to achieve. And I'm guessing, I'd put money on it, that if you look down that list, most of you, probably all of you, would be interested in more than one. Probably all of them. Many of them. So if you want many functions, many benefits, if you want to fix many aspects to your soil, it follows that you might need to use many different species. And that's the point about the cocktail covers or the diverse stands of plants. You've got different plants with different specialities, different modes of action, different functions that can do all of those different things all at the same time. Multifunctionality. So if you want multifunctionality, which is kind of a list of all of those or most of those, then that's where some of the diversity discussion can be very beneficial. So you've got, all that kind of list, and I'm not going to dwell on this. You know the deal. Legumes, there's kind of different ones we could choose from. Some of our broadleaf plants, you've got a whole range, brassicas and others, forbs, etc. Or we've got our monocots, the grasses, the cereals, and just put a mixture of those together. I'm not a cover crop expert for this part of the world. Someone else can tell you maybe a good mix of those and which ones and what rates and that kind of thing. That's not my department. That's you guys in your local context. But the point is they all do slightly different things. It's that diversity, different specialities, different rooting depths, different root exudates. Each one of these different plant species will produce different root exudates, stimulating different parts of that soil microbiome. And that's what we're trying to encourage, ultimately. So again, with that top-down view of soil health and acknowledging that plants and photosynthesis and roots, driving the roots, very direct benefits on changes. It's roots that decompact soil. It's roots that are cycling, stripping nutrients off the soil. It's roots that are feeding the soil biology. It's about how much more roots we can grow. And sure, diversity can give us a diversity of root biomass, of root exudates, of root structures, etc. And so I share this study. It's a really interesting one looking at, we often talk about diversity and the benefits of diversity, like I was just doing, to below ground, like this diverse, different roots, different feeding of biology, all of these great below ground things that we often talk about diversity. And I share this really interesting study looking at some cover crop, catch crops. I don't know if you guys use the word catch crops. It's kind of a thing over in Europe. It's basically a cover crop, but normally we would associate it as more of like a summer cover, like a really short temporary cover, kind of in the summertime between two of your main cash crops, like a cover crop in the winter kind of thing. It's basically very similar. But so we're comparing here a mustard, a monoculture, with a 4-way mix or a 12-way mix, and they're looking at a really interesting study done in the lab, that they're looking at how much carbon dioxide the plants, these different treatments, will breathe in. So they're looking at CO2 uptake from a mono of a mustard versus a 4-way mix and a 12-way mix. And they found here that carbon dioxide uptake was 2 times higher in the 4-way mix and 3 times higher in the 12-way mix. So we often talk about, and I'm a roots man. I love the below ground stuff and the root interactions and the root biome and root to root and all this kind of stuff. That's kind of my passion. And so we're going to get into more of those nitty-gritties in a minute. I love talking about all that kind of stuff. But here we're pointing out something really obvious and simple, that with that little bit more diverse stand, this was breathing in 3 times more carbon dioxide. And that carbon dioxide is the fuel of the whole system. Once we fix it into sugars and then plant biomass, it's the carbon, it's the energy that comes down into the soil to drive all of those changes we were just talking about, from soil biology to soil chemistry to soil physics. It's all about carbon and how much we can get into the soil. And so here we're pointing out that actually under some of those more diverse stands, we are increasing their ability to photosynthesize. All of the different plant heights, different leaf structures, more maybe upright or more out, they're capturing more sunlight, they're being more efficient at that photosynthetic process, which ultimately means more carbon into the system, which then drives all of those changes. So from that more carbon into the system, they found that the microbial biomass was 8% higher. Microbial growth response to that was 8% higher in the 4-way mix and 18% higher in the 12-way mix. Fungi and actobacteria were particularly responsive. And they also found that under the diverse mixtures, compared to the mono, that the carbon residence time, or how long that carbon lived in the soil or was stabilized in that soil, also increased in the mixtures by up to 1.8 times. So if I could just read this quote for you, the results of this study suggest positive impacts on plant diversity on carbon cycling by higher atmospheric carbon uptake, then higher transport rates of that carbon down towards the rhizosphere. higher microbial incorporation and prolonged residence time of that carbon in the soil environment. Okay, but it all started with more photosynthesis, more energy into the system, and then it just goes down into the system and then everything responds positively. It's really as simple as that. Okay, and then this is a grassland study for our grassland people here, looking at also some diverse mixtures of plants and its effects on soil carbon. I know this is an extremely busy and messy slide here. I'll summarize the key point to you. We're looking at the red, so it's our plant species here is 1, 2, 4, 8, and 16. And really we're comparing a whole bunch of things here from root carbon storage, the overall root biomass and how that influences soil carbon, kind of the soil carbon concentrations, even above ground carbon. And you can see across all of these, I won't go into the detail, but you can see in all of these, it's the red ones, which is the 16 species, that whether you're talking about soil carbon, root biomass, above ground biomass, it's very clear that the red treatments consistently, the 16 species mixes are consistently higher. So yes, more diverse plant species diversity also encourages more carbon accumulation in the system there. Okay, and so we talked a bit about the benefits of diversity there. Let's come back to just a few little 1 to 2 examples just to kind of bring it back to some cropping context too, because I know everyone can understand the concepts and the benefits of cocktails and diversity, but we still struggle to kind of integrate that into the cropping phase. And that's why I think that all the research that's happening on the intercropping space, just from 1 to 2, is really valuable because it shows you the beginnings of what's possible, the beginnings of that diversity, some of those diverse root-to-root interactions. And sure, maybe we don't have the technology to grow 5-way poly crops at the moment and easily separate them into 5 different crops. Sure, we can't do that yet, but hey, maybe one day. But there's plenty of enough good evidence to demonstrate that even 2 versus 1, it's really worth your effort. There's a lot of changes and gain that come from that. And a big part of that is, again, those root exudates that we've talked about. Here we have a mono cereal over here. It's only got the root exudates that it has, and it's pumping those out, and it can only communicate to the microbes that it can communicate to, that it can recruit, that it can turn on and off. And of course, those microbes that the cereal needs and uses and works with is unique and different to that plant. And that's different than to a legume or to a brassica or to anything else, some other broadleaf. They're all unique, they're all different, but they all have their sets of root exudates. They all communicate to a certain fraction, a certain portion of that soil microbiome. And that can be different from each of those different plant species. So when we put a legume in here with this mixture, now the legume is bringing some different root exudates, communicating with different microbes, waking up a whole bunch of different microbes that the cereal cannot do. And so when we intermingle those root systems and we have this companion or intercropping system where those root to roots are intermingling, now the cereal is benefiting from microbes in the soil that the legume turned on. The legume activated them, and now through this interface of interaction, the cereal can benefit from some of these microbes that it would not normally be able to interact with otherwise. And of course, vice versa. The legume is benefiting from some of the microbes that the cereal is inducing and activating. So therefore, this is a synergy. This is an interaction. This is 1 + 1 = 3. There is something, if we did a monoculture of cereal over here and studied that biome, and we did a monoculture of the legume over here and we studied that biome, and we've kind of classified, looked how much was there, what the composition, the community structure, all these things, we measure all these things there. If we studied them individually and then put them together and study them as one whole, we will find completely different things happening there as compared to studying them individually. And this is the essence of that problem with reductionist science. And hey, reductionist science has a lot of strengths. There's a lot of really good things about it, breaking everything down into its individual components. But you can't always translate from that back to the whole. That's the negative of reductionist science. It doesn't address the whole picture. There are interactions happening here in this interaction, 1 + 1 = 3. There are things happening there that you cannot observe in each individual monoculture. And that is the essence of why we do need a bit more holism, holistic thinking, holistic science, to try and unpack some of these interactions. Here's a great example. This is maize with some different companions. We're going to compare monoculture maize with a couple of different companions, soy, lupin, and mustard. And it was the lupin that was the one that had the most significant effect here. So what you're looking at here is we are measuring root exudate production from maize. Every single bar you see here is just measuring the root exudates from maize. It's just from what maize does. Now the brown one, there's 3 different types of different root exudates, all that are particularly important for scavenging phosphorus. So here we have in brown monoculture maize. This is the root exudates from monoculture maize of these 3 different types of root exudates. When we grow maize in monoculture, here's the level or the amount of root exudates that maize is producing. Now when we add a companion to that, and it was particularly lupin was the more significant effect here, and which is this green one here. Just by adding a lupin companion, I'm still measuring the root exudates from the maize. I'm not talking about what I did before saying, oh, the legume did all this stuff. Here I was talking about, oh, the legume did the work. The legume started activating all these others, and the cereal benefited from that. I'm not talking about this here. We're still measuring the exudates from the maize. This is exudates from maize as a mono. This is exudates from maize when there's a companion there, when there's a lupin there. And as you can see, in all 3 of these different types of root exudates, simply by having the companion there, the lupin there, the maize changed how it grows. It changed its behavior. It changed its root exudate profile. The maize produced more root exudates in response to having a lupin neighbor. And therefore, the phosphorus uptake in this maize that simply had a lupin companion was then increased. So it increased overall phosphorus uptake. So this is this interaction. If you study maize on its own, you can't observe this. You would say, oh no, this is the potential of what maize can do. Maize can only release this amount of root exudates. But have another companion there, and then maize interacts and changes what it does. And it increased its ability to scavenge phosphorus from that soil and be more efficient. Leverage that total locked up phosphorus that's there in the soil and make better use of it. Okay, so that's the idea, and that's really what that holism thing is about. Interaction, systems thinking, trying to study the whole system, the whole unit, rather than kind of always breaking things down into their individual little pieces. Okay, a little bit now, because I know you guys will appreciate this one. You're in a dry climate. Here we have a little bit on plant diversity and drought tolerance. This is also a really great study that basically was looking at this, saying how water use efficient are diverse stands versus monocultures. And I might just jump to kind of this one here to summarize this here. What they really point out is that different plant species, again, they're different. They have different specializations, different unique abilities, adaptations, etc. And some plants have very different ways in which they tolerate dry conditions. They have unique adaptations to handle dry conditions. And so, for example, this kind of green one here, this first green one, this is dehydration avoidance, meaning that once that plant is established and has moisture, it's really good at holding on to moisture. So as you then move through drought conditions, so as you move over time, its unique ability is to simply hold on to the moisture that it's got and maintain growth and maintain production in a drying soil, because it's very good at keeping all that water back, locking it up inside itself and holding on to that. And so, yeah, sure, over time, once it does eventually start to lose that moisture, okay, its productivity is beginning to decline. Its real specialization was up here when it was holding on to moisture, and then sure, over time that declines. But for example, you have other plants that have different mechanisms of dealing with dry. For example, dehydration tolerance. And what you can see here is the opposite, where, okay, what this plant is very good at is just growing in dry conditions. This plant's actually not so good at drying in dry conditions, but it's just good at holding on to moisture if it got it at the beginning in the spring, say. But you have the other one that's actually good at maintaining production in dry soils. So if you just had one or the other here, if you had a mono of one of these other, yeah, initially you'd have really good biomass production if you had this one. But as the drought extends through time and gets more and more severe, you're really losing out. Whereas if you just had this one, you wouldn't be optimizing the efficiency of that kind of earlier or earlier moist conditions. You're also just, okay, you're getting the better deal at the back end versus maybe the front end. So if you bring the two together, then you get the best of both worlds. One that's going to be really good production in the early stages of drought, and another that's going to be really good at production in the latter stages of drought. So if you had one or the other, you're not optimizing both of those specialities or those unique abilities to then overall maintain increased productivity. And so from a moisture point of view, there's other studies I could share with you. There's quite a few that have looked at dry conditions, and diversity also really helps with tolerating dry conditions. And also in an intercrop, companion crop could also be. Here's a really great study. This was just a pot trial, a little lab trial growing in pots, as you can see here. And we have a deep-rooted legume. This is pigeon pea. And then we have a finger millet. And they're both growing together in this pot. We have a water well down the bottom. And what they did is they watered from the top, and they got these two plants established. And you can see here that the finger millet is growing in a little very fine mesh bag, which means there is no root-to-root interactions. They are not connecting. They are not interacting. There's no connection there. This is completely blocking off root activity, root connectivity. So they water and they get everything established, and then they stop watering. So they impose a drought from the top. And so, of course, poor old finger millet, who's not a deep-rooted plant, he can't get down to this water well here like our pigeon pea can. And the pigeon pea maintains production, but we see the finger millet really stop right down under those drying conditions. But then, on this treatment over here, they added mycorrhizal fungi. And mycorrhizal fungi are much more finer than plant roots, so those fungal hyphae can penetrate through this mesh barrier. So now the two plants are connected through the mycorrhizal fungi. So then again, they impose a drought from the top, and our pigeon pea, turns out it then will access that moisture, and it shares it through the mycorrhizal fungi through there, and we see increases in finger millet production. So even in this example, the diverse stand can actually increase your water use efficiency, increase water access, and overall increase biomass production. So it can be that the bit of diversity can also pay other benefits and dividends there. So in that context of intercropping, I mean, again, I kind of break it down into some of these basics. Really, legumes are typically the centerpiece of a good intercrop or companion crop. And that's, of course, legumes are very good at the nitrogen thing, of course. But legumes are also very good. They have very good acidic root exudates. They're very good nutrient scavengers. They are very good at scavenging phosphorus from the soil, calcium from the soil. Of course, they bring that nitrogen in as well. So they're not such a hungry crop. They have a low nitrogen demand, whereas cereals have a high nitrogen demand. Typically, our oilseeds have a high nitrogen demand. So we want to bring together that complementary nature. So they're not going to demand so much. They can feed themselves, which means they're going to have stuff to share with the others. So really, in terms of a companion, it's limited only by your imagination. But generally, you would say, okay, you could pretty much combine one of these legumes with any one of these cereals or any one of these oilseeds, like whatever's relevant for your context and that kind of thing. And you can do all sorts. I mean, you know, oats and peas, that's like one of the classic ones that a lot of our forefathers and grandparents used to do. I mean, oats and peas is just a classic one. Barley and peas equally. I mean, wheat and beans is a really good one up in the Canadian prairies. Chickpeas and flax is become super common. Lentils and flax is another one quite well. Of course, peas and canola, you know, that's the other one, of course, as well. Limited by whatever. And sure, look, again, it's easy to say. I know there's specifics underneath that of some of the technical side of managing some of the herbicides, the rotation. Like there's some of these agronomic issues. I'm not saying it's a walk in the park. You've got to deal with separating, et cetera, et cetera. But if anyone's interested, like I would encourage you to have a look to the prairies up in Canada there. They're really embracing kind of some of this. There's lots of good stuff happening there in this space. And despite the hassle of having to separate it post-harvest, farmers there feel it's worth it. Even though you've got the cost and the labor and the equipment to separate, the benefits that they see in field in terms of nitrogen reductions, disease suppression. This is the big one for chickpeas and flax. Chickpeas are very prone to disease. With this chickpea flax, there's finding much lower fungicide use, lower disease pressure. There are economic benefits that pay in the season that ultimately make that worthwhile. So I think, yeah, it's just that idea of different plants, different things, different specialities, bringing them together. Here's another example of just pH. Here we have fava bean. Look at the pH of the rhizosphere. It's really acidic, you can see. Here we have maize. Look at the rhizosphere. It's a bit more neutral. So again, different plants, they release different root exudates, recruit different microbes. And so when you bring these together, there's that opportunity there for synergy, for interaction. And even think about the maize. Like here we've got acidic conditions, like some of the trace minerals and things are really nice and available here. So that maize, it's a bit more neutral, as it sends its roots over here and they intermingle. That maize can kind of get some of the trace elements from that very acidic root zone. Or equally, the legume can get things like mole or calcium or magnesium from this root zone that are a bit more available. So again, there's this kind of basic kind of sharing resources, even at a basic pH level, but of course we could extend it well beyond that. So a couple of quick examples to close. We saw this one already. There's your alternate row intercrop. Now you can also do the all-in-one line. That's your kind of mixed row, just mixing them all together. And that's also okay as well. I'm going to show you an example of this in a second. But there's a nice example of that interrow. Of course, you've got your, this was the one I mentioned earlier. This is a temporary intercrop or the companion crop. And here you can see this is canola. You see down here with a ginormous stand of beans, which they were both planted in the autumn. This is from the UK, planted autumn. So grow together, and then come the spring when everything's kicking off and breaking dormancy, the farmer then, with a selective herbicide, terminated the beans. Of course, those beans are a legume full of nitrogen. Now they are decaying and breaking down and feeding the canola so that it can go then through and finish off through to harvest. The farmer did acknowledge that he sprayed off a little later than he would have liked. They're probably a bit too competing with the canola there that you can see. But nonetheless, there's a good example of this fantastic synergy of root-to-root interactions during those kind of vegetative and winter stage, and then come spring, when you want to finish off the crop, terminate the other, let it decay and feed the canola and take it through to harvest. Then you've of course got the relay. This is kind of most famous from your country, of course, with things like the relay intercropping, spatial, again, bringing a bit of that spatial separation, but overlapping with relay. Also some really good example happening there. And just to say, on the nitrogen, on the legume, non-legume companions and intercrops, yes, we have the evidence. We know that legumes can share nitrogen in real time during that growing season. It's not just when they die and decay and then release that nitrogen for the next crop in the rotation. If you can marry up a legume companion with your cereal or with your oilseed, whatever, the legume will share nitrogen to the non-legume. And the two pathways in real time, apart from the death and decay pathway, the two pathways is one through just root exudates. We talked about that already. The three main classes of root exudates are sugars, carbohydrates, amino acids, and organic acids. Amino acids, organic forms of nitrogen that are excreting out of the root system as root exudates. So when you have root-to-root intermingling with a nitrogen-hungry crop like a cereal, then it is going to scavenge those amino acids from the root zone of the legume. And not only does it do this, this actually forces the legume to keep on fixing more nitrogen. So a legume with a non-legume companion will fix more nitrogen than a legume monoculture. And we have many studies that show this, for the very simple reason that that cereal, that non-legume, keeps stealing the nitrogen. So the nitrogen never builds up enough around the legume for that nitrate to build up, for that signal that we talked about, for the signal to say, okay, there's enough nitrogen here around you, don't bother fixing more nitrogen. The legume never gets that signal because the non-legume keeps depleting the soil nitrogen, taking it up, taking it up, taking it up. So the legume keeps on fixing nitrogen. You will fix more nitrogen with a legume with a companion than you will with a monoculture legume because of that reason. So we see the sharing of root exudates. However, in order to optimize that process, you need the right varieties or the right plant species which have particularly lateral roots. If you have a plant like alfalfa, that's a great nitrogen fixer, but it has very deep roots, it's actually not so good at sharing nitrogen. You've got to have plants or varieties that have more lateral roots where they can then interact. So we have good examples of this. So it can be down to a varietal thing. And of course, we have not bred for this in the last 50 years of plant breeding. No one's bred for this. So sure, maybe the amount of nitrogen shared isn't significant. It can be reasonable, but it's not like you're not going to fall off your chair with how much free nitrogen you can get, but you can get some. But we haven't bred for that. What if we bred our varieties to have these lateral roots and to share that nitrogen? So for example, we do know that in the example of clover, little leaf clovers will share more nitrogen than big leaf clovers varieties, because a big leaf clover variety needs to hold more nitrogen back to grow that big leaf. So it doesn't share as much. A little leaf, microclover, has a small leaf, so it can share a lot more. So we could breed for this and breed legumes to be companions. What kind of varieties do we use in legumes at the moment? We use varieties that have been bred to be productive under monocultures. They're not the right varieties. I mean, don't get me wrong, they work already, but they have not been fine-tuned for this kind of a process. So if we bred them to be companions, we could get a whole lot more free nitrogen from this kind of interaction. So that's Rutexudates, and then there's mycorrhizal fungi. Only if the two plants are mycorrhizal associating, so you won't get this with a pea canola, for example. It has to be a mycorrhizal plant, so yeah, any of the cereals or grasses. So some of that nitrogen fix will actually be passed directly through mycorrhizal fungi into the non-legume. So yes, we can also see nitrogen sharing in real time in those pathways. And again, that's an opportunity for nitrogen reductions. Couple that with some foliar sprays of urea, keeping soil levels down. Yeah, as a system, we can start to piece these things together and have meaningful impact on our reducing inputs. So here's just an example of alternate rows versus all-in-one, or kind of more mixed, alternate row intercropping versus mixed. And you can see here we have peas, your other peas and the classic beans. And you can see the alternate row kind of patterns versus all mixed. Now, the alternate row patterns certainly helps a bit more so with lowering disease pressure. But in terms of the nitrogen sharing, it seems to be maybe a bit better that they're all mixed in one. But to be honest, farmers have kind of been playing around with both of these from what I see, and either way, it still seems to be a lot of benefits to those. So I think whichever way you do it, I think is fine, but there's a bit of nuance there. Again, like just the absolute classic pea and oats, that's all just mixed together. You don't have to have the machinery and kind of separate out those alternate rows, but sure, if you can, there's benefits there, but if not, put all the seed in one. I think that's a nice example, one of those classic examples. There's another one there as well, barley and peas. So, okay, I just wanted to share a couple of those little examples of in the cropping world, where even just from 1 to 2, there are some benefits to changes in root exudation, changes in soil biology, changes in soil carbon, changes in phosphorus scavenging, changes in nitrogen use, nutrient sharing, etc., etc. There are lots of other benefits that kind of come from that too. But in summary, of course, yeah, look, diversity is great. I really support it. I could talk probably a whole another hour about it and show you all sorts of other things about how it can improve aggregation and soil structure and other aspects of nutrient cycling, the disease pressures and things that we talked about earlier. There's so much there to kind of talk about and say. So yeah, sure. However you can apply it and integrate it, I support diversity. I think it's great. But also, I know this was maize, and we had a companion understory here with some ryegrass, but now that maize is gone. And if I look at this here, this is a monoculture of ryegrass. And in my book, there is nothing wrong with that compared to that. So yes, diversity is great. I love diversity. I support it. But if I had a choice right here, I would absolutely have no problem with this monoculture. It's keeping that soil covered. It's capturing the last of that fall sunlight. It's obviously maintaining soil structure there so that we don't have this poor infiltration, water puddling there, etc., etc. Green leaf, photosynthesis, carbon energy coming down. There's absolutely nothing wrong with this. Okay? I'm not hardcore anti-monoculture guy, although I do think the diversity thing is way better, and I really support it. And sure, why not? Couldn't we just throw in like at least maybe one legume there? What about some grass clover? Even better. Maybe a brassica, maybe some other legume, whatever. You know, sure, a little bit extra diversity. 3 or 4 species, amazing. Sure, if we could, I'd be really happy with that too. But I just want to be grounded in that and just say, look, compared to nothing, compared to zero soil cover, I would rather have this 1 versus none. As good as I think 2 versus 1 are, I'm also cool with 1 if the alternative is none. So nothing wrong with that. I still think the soil health principle of living cover, living roots, keep that green cover is still the foundation, the absolute key. And sure, beyond that, if you want to dabble in the diversity thing and play around with all of that, also has my vote. I think that's also fantastic. Okay. Questions? We got there. We made it. You all did well. We got to the end of the day. I know I'm a little over time. It's 20 past 4, so if anyone does need to urgently rush off, feel free to quietly do so. But if anyone wants to take a few questions, then happy to take a couple of quick questions. Yeah. A regenerative style pragmatism? Oh, practice. Sure, in the sense. Sorry. Sure, in the sense of course, before the maize was harvested, you know, we had a nice little companion crop going on here. You know, so yeah, I would consider that's great, and that fits within that philosophy. But you know, it's still a good practice, irrespective of the regen label. You know, and I think that, like again, classic oats and peas that our grandparents used to do, like it was just a good practice in that day. And whether or not that came with that label or not, sure, I just think it's a good management practice. And sure, yeah, well, sure, yeah, most definitely. Mhm. Yes, and I think that if you started here and you started to see these kind of differences, I'm pretty sure you're going to start to be a little bit interested in what else comes down further down the path. So I think it's a great stepping stone, great beginner, great entry point for sure. Yeah. Okay. Mhm. Mhm. Yes, for sure. Absolutely. Yeah, yeah. So the question there was, you know, are there some annuals that are still good, that do have good root systems? And yeah, you're right, absolutely. And again, I used the example there in the lecture of the some of the heritage wheats. You know, they do have distinctly. I've got a really great photo of it. I didn't use it today, but distinctly much deeper root system. So although they're annuals, on the scale of annuals, they're at that top end of being annual, but still producing lots of good roots. So yes, even within an annual system, if you can steer towards some of those annuals that do have those good rooting characteristics, the better. And again, that point about plant breeding that I just made about breeding legumes for nitrogen sharing, we can breed annuals to have better root systems. And again, this could be that point of what I call cover crops 2.0. It's the same argument about your cover crops. The varieties you're using for most of your cover crops are varieties that have been bred for their productive goals, which may not make them the best at being a cover crop for soil restoration. So my view is that cover crops 2.0, we will see specific varieties bred to be cover crops, and most times that's going to mean more roots, in my view. So yes, we definitely could be breeding our modern varieties to have a bit better rooting characteristics. And there is some of that work happening now, looking at kind of crossing modern with some of those older ones, or just going back to those older ones and seeing what else we can, which other directions we can take them. Yeah. Okay. Cool. Thank you very much. Is there anything there you need to?
We'll quickly do our door prize drawing.